because of the focus of this series, I thought I could really talk about imaging cognitive reserve. And really, a lot of this is sort of a personal history of how I began to think about using imaging and how I'm thinking about it now. So I'll be really interested in the discussion subsequently, and I hope I can keep it short enough that we have a lot of time for discussion. Let me get this on here. Okay, everything sounds better in French. Cognitive reserve, qu'est-ce que c'est? Sort of like psycho killer, qu'est-ce que c'est? What is reserve? So now we think of reserve as something that moderates between brain changes and their clinical manifestations. Here it's drawn as a moderator. We can talk a little bit about the discussions lately about whether pure moderation is necessary or not. Here's the white paper definition for those who are not familiar. This is from the PIA, from the AAIC. We have a reserve and resilience PIA, and this was sort of the sum opinions of 30-odd people. And cognitive reserve refers to the adaptability of cognitive processes. It helps to explain differential susceptibility of cognitive abilities or day-to-day function to brain aging, pathology, or insult. And notice what we added in there, adaptability, i.e. efficiency, capacity, flexibility. And so those are some of the kinds of issues that I want to talk about, because those are the kinds of things that we can use imaging to look at. The epidemiologic evidence for reserve, I'll summarize very briefly. Proxies for reserve like IQ, educational literacy, occupational attainment, leisure activity, social networks, etc., have been associated with reduced risk of incident dementia and more rapid cognitive decline and mortality in AD. Notice I put the last bullet in parentheses, slower rate of cognitive decline in normal aging, because there's really disparate findings on this. In two different studies in our North Manhattan cohort, we were able to show slower rate of cognitive decline, even using sort of the more modern statistical techniques, but other people haven't seen it. And I have ideas of why that is. What I'll show later, and many of us have shown, is that even in normal aging, we can show reserve moderating between brain and cognition. So here's the very first paper I ever published on reserve, and it's the first imaging study. This was with xenon, which is a blood flow measure. And here what we showed is that now I know what to call it. This concept of neurodegeneration is now prevalent in AD, where first you have amyloid, then tau, then neurodegeneration, which is seen either as cortical thickness changes or metabolic changes. So here we were measuring brain metabolism, and we showed that we had three groups of patients, high, intermediate, and low education. Controlling for disease severity, the people with high education had greater depletion in this temporoparietal area than the other two groups. Suggesting that even though they looked the same clinically, they were walking around with more AD pathology. And I like this figure because it illustrates key components of studying cognitive reserve: brain pathology, so measure, clinical outcome, and the moderator. And this incorporates all of that. So I was very excited with this finding. And it's been replicated numerous times now with more up-to-date measures of pathology, better measures of neurodegeneration, et cetera. You might not know that we did the same study looking at occupational attainment. Here and what I really thought is that higher occupations with greater cognitive challenge would impart reserve. But what we found is that two features of occupation, interpersonal skills and physical demands, were the ones that imparted more reserve over and above education. So you can see how we did it. We had age, we're controlling for age, mini-mental, that's cognition, function, duration, education. We were counting for 30% of the variance. When we added interpersonal skills and then physical demands, we accounted for even more of the variance. This figure is for interpersonal skills. Another thing I could mention is that when I show these and when we first saw them here and here, you can see that we show this greater depletion in the people with the higher reserve proxies, but we also seem to show greater flow in the frontal areas. Question is that compensation or not? That was a very long debate between us when we were publishing these papers, but we did not make that claim. And we could talk about why. much later, when we already were doing O15 PET, we did a similar analysis now looking at leisure activities, and again showed that controlling for disease severity, the higher the reserve, the more the depletion in these areas highlighted in red. So those were very early imaging studies, not functional imaging studies. So I was convinced that cognitive reserve was a real thing and very itching to understand, how does it work in the brain? How's it neurally implemented? And I think I was seduced into using functional imaging because that was very hot at the time. When I started thinking about this, we didn't have fMRI yet. We did have O15 PET. And the idea was to understand how cognitive reserve can be neurally implemented. So it's a much more complex problem. We're not interested in the tasks directly that are involved in task performance. We're interested in networks that are mediating cognitive reserve. So, we always have that problem in cognitive reserve studies where we're not just interested in what underlies performance, we're looking at individual differences and studying individual differences. So early on, simplistically, cognitive reserve operates through individual differences in how tasks are processed in the brain. So early on, I thought a simple approach would be to challenge participants with the task and investigate differences in task-related activation between individuals with high and low cognitive reserve. It sounds very simple, but that's really the way that I chose to start. This last bullet is a sort of a funny story, but I'll say it because I had a colleague who really could not understand how I was thinking about this. And I think that the confusion was that he felt that if I wanted to understand cognitive reserve, I should go for ascertaining networks that I thought underlie reserve. Can I find this true source of reserve? But what I was assuming is because cognitive reserve modulates most aspects of cognitive performance in the presence of brain change or pathology, we can take any demanding task and get some insight from it. So very early on, I published this quite a few times. It might look a little reminiscent of Crunch, but this was way before Crunch, and we're not showing any Crunch. But this was something that the issue that was I was obsessed with in the beginning, and that is if we increase task demand, we'll certainly typically show increase in activation, task-related activation, and there might be some inherent differences between young and old people. So what I'm showing here is as the task gets higher, because of less efficiency, older people might ramp up a network more rapidly than younger people. And then because of capacity, older people might have less capacity, so they'll top out at a much lower level than young people who have much greater capacity. So that what I was really concerned about is that if we give two groups, let's say young and old or old and AD, a task, we might see differences that are purely a function of efficiency and capacity and not truly related to compensation. And there were studies that were coming out then that made that clear. So one that you might recall, I didn't put up any slides on it, Cheryl Grady had a study. She was a real pioneer in the whole concept of compensation. And she had a study looking at a face matching task, I believe it was, and she showed that older people showed greater flow activation in some frontal areas than younger people. And she hypothesized that that might be compensation for aging. But subsequently, she published a paper where she used degraded faces, and she showed that young people started to activate that same area. So for me, that's not a compensation effect, that's a difficulty effect. And I think those are two different things that are important to distinguish between. We can discuss it more in the Q&A. So I was really looking for approaches to control for difficulty, or at least take difficulty into account. Just a little sidebar, here's this was a lot of work, this study, so I just want to show it. Given these efficiency capacity curves, we try to do our own study to check that out. It's a cool study. We show people two of these loopy shapes. They get a probe where they have to say yes or no, that was one of the two shapes. But what happens is a mask comes on immediately following the probe. And the deal is, or at some point following the probe, so it could be just 125 milliseconds after the probe, 250, 500, 1000, or 2000. So we're modulating difficulty by giving people less and less time to make their response. And what we have in terms of discriminability, you can see in the, here's the net amount of time that we give people to do the task, to make the decision. So the less time they have, the worse they do in terms of discriminability. As they have more time, they do better. And you can see that the young people ramp up that ability much more quickly than the old, this dotted line is old, and have higher capacity as well. And what we were able to show is we found a covariance pattern that increased with task demand in both groups. And we could show that when the task was easy, the older people in blue were actually using that pattern, activating that pattern more than the young, to me, lower efficiency. And then when the task was very hard, the younger people were ramping up that network more than. So this issue of task difficulty and demand was something that was bugging me. And so our earliest studies were with O15 PET, and this is what I decided to do. We decided to go for a titrated demand list learning. So what we would do is show people shapes. Here's a list, study list size of 1. We'd show a shape. If it had a box around it, they had to say, was that one of the shapes I just saw? They could say yes, no. Show them a new shape, yes, no, a new shape, yes, no. If the study list size is 3, they'd see 1, 2, 3 shapes, then they would be queried 3 times, and then they get 3 more shapes. So what we decided to do was usually pit study list size 1 against a titrated demand, where before we put people in the scanner, we actually varied the study list size so that every single subject in the titrated demand was performing at 75% accuracy. And I felt that once I controlled for this difficulty, differences that I would see between young and old people would be more likely related to compensation than just to different efficiency capacity. In other words, just a difficulty effect. So we did, and this we did in O15 PET. I don't know if all of you are familiar there. You can do 1 task at a time for about a minute, then you wait 15 minutes, they get another injection of O15 PET, then you do a different task. So they'd have a session with study list size of 1 and a session with the titrated difficulty or however we set up the experiment. So this is not the first study we published using that approach, but it's one that I like because it made us think about some concepts. So here with these are all working with Chris Habeck, who, because we work with these covariance patterns. So here, what we did is we pitted set size 1 against titrated set size, and we found a covariance pattern whose increase in expression from set size 1 to titrated demand differed across the young and old. So you can see the young, as they had greater cognitive reserve, according to our cognitive reserve factor, expressed this network more from low to titrated demand. The old people expressed it less. So we could say, oh yeah, the old people must be using this network in a different way than the young people. And that led us to these concepts of the neural implementation of reserve. And I feel a little badly about this paper because we coined these terms that are really maladroit. So the reviewer said, well, you should call them neural because you're talking about the neural. And I think the mistake was to call one of them neural reserve. So what I'm talking about is the neural underpinning of cognitive reserve. One of them is neural reserve. And the neural reserve represents normally occurring individual differences in capacity to perform tasks or cope with increases in task difficulty. These differences could result from innate differences or exposures and might be characterized in terms of differential efficiency or capacity. That's an easy one, but could be just pre-existing differences in flexibility of solution strategies or the like. So old and young people could have networks that subserve neural reserve, people with pathology can, and then when they're challenged in some way by a task or pathology, those with higher neural reserve can better maintain performance. And then we call the other neural implementation of cognitive reserve neural compensation. the use of alternate networks not normally used as a response to disruption of normal networks, reorganizing the typical brain areas or recruitment of alternate brain areas. So we were claiming that here we were seeing neural compensation in that the older people were using that same set of brain areas, but clearly in a different way than the young. So this neural reserve and neural compensation, there's other concepts that are similar. Compensatory reallocation, we could call neural compensation. The Harold might be called neural compensation. But you can see why this thinking about these concepts is why I had not the greatest reaction to the Montreal paper, where the term compensation was used to encompass both efficiency, capacity, and compensation, because I think they're very important things that are worth differentiating. Okay, so what happened after a while is people in my lab convinced me that even if you try to titrate task difficulty using any kind of format, there's still going to be important differences in the task between young and old. So it's almost hopeful to say that we can equate task difficulty. So what we started to think about is just varying task difficulty and looking at change in task difficulty. And right about that time, there were a whole slew of papers coming out about the Letter-Sternberg task, the delayed match to sample task in the scanner. And I was very intrigued with it because if you're familiar with the task, people see 1, 3, or 6 letters. After a waiting period, they're probed with a letter and they have to say, oh, that was one of the letters that I saw, or it was not one of the letters that I saw. And there was very nice behavioral features to this. Reaction time systematically increases with set size in almost linear fashion. What I liked about the task wasn't that it was a study of working memory so much, but that here, just by varying one presentation, we can manipulate demand. So the task remains very constant, but we can manipulate demand just by increasing set size here. And so what you're typically interested in was load-related activation. The change in activation as set size increases, and we focus on that because cognitive reserve might be more related to the coping with increases in task demand than task-specific features. So we've done a lot of work with this task. Here's the very earliest, I think it's our first fMRI study using this task. This was with Chris, again, and just 40 young adults. Chris, using the scaled subprofile modeling, isolated an activation network that was expressed across all 4 lows, set size 1, 3, and 6, but typically increased in expression as the task got harder, what he calls an ordinal trends approach. And what we were able to show, everything that looked very nice here, is that in these young people, those who ramped that network up more had worse reaction times than those who ramped up the network less. And also those who ramped up the network less had higher IQ than those who ramped up the network more, as if they had greater efficiency. So it was a nice, clean finding. We're having trouble replicating this with N's of 120, but it worked well with 40 people. And then subsequently, we had just 40 young and 18 old subjects, exact same tasks, exact same measures. And here was a whole series of analyses. Here we were working with Eric Zarin, who was in our lab at the time, and he used a covariance analytic approach called MLM, multivariate linear modeling. So I've presented this a lot, but let me walk you through it in some detail. So we looked at load-related activation, in other words, a network that increased in expression here during encoding. And what we found is that both the young and old used a set of brain areas that are typically associated with working memory, but greater expression in the old than the young. Greater expression here is down for measurement reasons. So on average, the older people are using this network more than the young. So this we could talk about in terms of neural reserve or inefficiency. This network seems to be less efficient in the old. Then came the next finding. There was a second pattern that we saw with MLM. And this pattern was primarily expressed in the elders. See, here's the open squares are the young, and you see their results hover around 0. But you can see that the dark squares are the elder. It's the elder, our expression of this are different than 0. So you'd say, oh, cool, that's a compensatory network that the older people are using. But what we found is that higher expression of the pattern was associated with poorer performance. So this led to a little debate within the lab. So Eric was saying, look, that can't be compensation. Compensation is associated with better performance. And here it's with worse performance. I'd say that we should label this de-differentiation, which is a popular term I'm sure you're all familiar with. I argued that, well, it still could be compensation. It could be that once that primary network, this network here, loses efficiency, maybe the older people resort to another network that allows them to do the task, but not as well. Like a cane is compensatory. You can still walk, but not as well as you could before. And if you read this 2007 paper, knowing that background, you'll have a lot of fun because he wasn't thrilled with that idea, but I was the PI, so these concepts were discussed in a very ginger fashion. That's the intermediate, the social part of doing research. Along comes Jason Steffener, who was a postdoc later, worked with me for quite a while, and now is on his own in Ottawa. So Jason really took that very seriously. So is this expression of pattern to de-differentiation and neural compensation? So it's very hard to see here, but what he did is in outlining in black, in the brain, he outlined the areas of the first network, and he looked at gray matter density. This was before Free Surfer, and he found a spot, you can see it here, in the left SMA, where gray matter density was increased, where decreasing gray matter density was related to increase, that's in pattern 1, in a part of the brain associated with pattern 1, was associated with greater expression of pattern 2. So we had a direct evidence that somehow when pattern 1 was affected by some change in the brain, these subjects were expressing pattern 2. And this would suggest that pattern 2 is really compensatory in helping maintain performance. So this was a long set of papers. And then Jason went on to do some path analyses that have really influenced how we do our research to this day. And these are really based on a true path analysis. So here, age-related pathology, in this case, it was gray matter density within a network can affect the efficiency of the network. And perhaps when efficiency becomes low, induce someone to use a compensatory network. And that neural activity then is related to task performance. And how can cognitive reserve be measured here? What Jason was able to show is using proxies of cognitive reserve, people could tolerate more of that gray matter density change and still maintain utility and efficiency of that first network without the need to compensate. The cognitive reserve is allowing people to maintain efficiency in the more beneficial network, or when it's decreased, people move to a compensatory network. So that was really a very nice story about cognitive reserve. The other thing that Jason found that influenced us going forward was this bar here. No matter what we saw in terms of task-related activation, we could see that our reserve proxy still moderated between task-related activation and performance, such that whatever's going on here, people with higher reserve still did a little better on the task. And that's the observation that led me to think of these task invariant reserve networks. In other words, what we're looking here is at task-related activation. Could there be other task invariant networks that allow people to do better, even though the task-related network might be affected? So a task invariant network, the logic behind it is cognitive reserve allows people to better maintain function in multiple activities and cognitive domains in the face of brain changes. Think of Alzheimer's disease. It's not just specific to memory reserve, it's people can cope with the disease much longer until it's expressed. So can we identify a pattern of cognitive reserve related brain activity in our first experiment common to two different tasks, independent of the network specifics to each task, and expression correlates with IQ? And that's what we did. We took advantage of the fact that we had measures of people on two. We had the letter Sternberg, the letter delayed match to sample that I was showing you, but we also used a delayed match to sample that used the shape stimuli. which are much harder and really have much different cognitive demands since it's a purely visual, nonverbal task. And what we were able to do is fish out a network, again using multivariate linear modeling, that was identified during the encoding phase of both tasks, and whose expression increased with higher IQ. And expression of this network was independent of task performance. So in the context of these tasks, this network represented a neural instantiation of cognitive reserve. We found it only in young people. The fact that it was very frontally based influenced me to start incorporating some executive tasks into our studies, but that was our first foray into a task invariant network. Subsequently, because we have another study where we can look at this more intensively, we have a study where in the referenceability neural network study, we're giving people 12 different tasks in the scanner. So we revisited this idea, and can we identify a cognitive reserve related brain pattern that's commonly expressed in 12 different tasks that have different processing demands, whose expression during performance correlates with the CR proxy, and whose utilization moderates the relationship between structural brain measures and task performance. This was just recently published, not so recently anymore, 2018. So we did find a network. This network was expressed, this is sort of a summary, again, it's a covariance pattern. In all 12 tasks, its expression correlated with IQ. I'm not showing it here, but we forward applied this pattern to a totally different task and showed even in that other task, if we looked at that pattern, its expression correlated with IQ. And then we could show that expression of that pattern moderated between cortical thickness and fluid reasoning. So we had an interaction effect such that as thickness was reduced, you can see the people with higher expression could maintain performance better than the people with low. We consider this the real test of, is it a reserve-like network? And then subsequently, we and many others, Nico and Michael for sure, have looked at resting bold connectome. So here was our attempt. We tried to find a resting bold connectome whose expression correlated with IQ. We identified one. So this was just using using just a set of ROIs without any a priori idea of what this should be. And then we tried to see whether we could find moderating effects, again, between cortical thickness and cognition. And what we found, both in the derivation sample and in a forward application to a new sample, was that the connectome score certainly accounted for better performance over and above thickness in both, but we did not find an interaction. We did not find moderation. We did find moderation in some of the individual tasks. We had 4 different cognitive domains, so we found some in some of the domains, but they weren't consistent across. The moderation wasn't consistent across the two different cohorts that we were. So that's why, you know, this is in revision. We're just sort of having to back off of our claims a little. Maybe someone listening to this is the reviewer. But yeah, so. And this led to a whole discussion. You know, we had considered moderation the sine qua non of identifying reserve, but talking to a bunch of my colleagues and a bunch of epidemiologists, they're leery of making that requirement, because it turns out that very often, if you just sort of scale some of your variables, you can create moderation. So this idea of showing additional benefit over and above thickness, many people now would feel is still reminiscent of reserve. So all of this has guided what we're doing now. So I'll just tell you what we're doing now. We published it in this special issue that on reserve that Oziyama edited. So we have brain morphology, cognitive performance, and here we have fMRI, and we have our reserve proxies. We're giving the delayed match to sample task and a set switching task. And now we can really do something that I hadn't done before, in great detail, measure all of the features of the brain: gray matter volume and cortical thickness, white matter hyperintensity burden, tract integrity, resting CVF, I really could put it over here, amyloid and tau burden. So in other words, we can look at this all given the background of the brain, which Jason's study and analyses made clear to me. So if you want to look at task-related activation and how it relates to cognitive performance, and you want to make a claim that differential task activation is related to cognitive reserve, it really should be in the presence of similar brain status, what we might call brain reserve, or just call it brain status. Better. So we have a lot of data on this in a study, people 20 to 30 and 60 to 70. And then what we have now is follow-up data. So now we can look at change in morphology, change in task-related expression, change in outcome, et cetera. Again, relating this to reserve. And given the next point that I'm going to make about our pilot studies, our pilot study request, we have longitudinal imaging data. Our longitudinal study was interrupted, but we do have longitudinal data with all of these measures, including task-related activation on our set switching and delayed match disconnect. So these are sort of just quickie conclusions, observations. I think imaging studies can help us clarify the neural implementation of reserve. And this neural reserve, neural compensation, I like to think of things that way. And then a general reserve network, what do we really call that in this context? I'm not really sure. In some way, I would place it in neural reserve. I think it's important to take into account measures of brain status, if you want to call that, it's what I call brain reserve these days. Differentiating compensation from efficiency capacity can be challenging. I think that the proper cognitive experimental studies that really highlight the underlying processes. So let's talk about strategy selection. Can we find a task that, let's say, has two different strategies, and can we get the neural signature of those two strategies? If we can do that, then we can really more finely study the kind of issues that we're interested in. And let me just conclude quickly about talking about this collaboratory for definitions on reserve and resilience. This is an NIA supported project. We had our first meeting last year. We're going to have our second meeting online in September. I see a lot of names of people who were there. The thing that we're doing now that I want to call your attention to is we have a call for pilot studies to validate and clarify definitions and concepts. It's just a 3-page proposal. Here's the link for it, reserveandresilience.com, call for pilot projects. It's due in the middle of this month. But the whole point of this is to have people take either longitudinal data, epidemiologic data, animal data, imaging data, create operational definitions for cognitive reserve, brain maintenance, et cetera, and show how they're testing those operational definitions in a project. We have posted on the website people who are willing to share their data or actually do these large cohorts. As I noted, we have a lot of data from the B.R.A.I.N study and from our cognitive reserve study, and we're certainly willing to share data. So for those of you who haven't contemplated one, it's just a quickie 3-page proposal. I really urge you to apply. And the next meeting that we're going to have is going to be mainly centered around these operational definitions and the accepted pilot projects, because I think that's the a good way to start to get to more brass tacks about how to define reserve and resilience. Okay, so I'll stop there. I haven't looked at the time. Let's see how I did. Pretty good, 40 minutes.
Yeah, thank you very much, Yaakov. That was fantastic. So please, everyone, come forward and just start asking questions, and go ahead and unmute yourself. So maybe okay, that's the first one.
Sorry. Hi, Yaakov. That was a really interesting talk. It's nice to get more clarification on it. I just wanted to ask: was there any overlap between the regions that came out in the task invariant network and the connectome, the resting state connectome?
That's a really good question, Rory. We haven't looked at that carefully. I don't know if Ada is on. She felt that some of the areas in our task invariant network seem to be related are reminiscent of a resting mold, I'm sorry, something that she saw on PET. Yeah, we haven't, but that's something we thought about, but we haven't looked at carefully yet.
Thanks very much. Thank you.
And
Okay.
here's one that popped up. What do you think about the role of diffusion imaging, white matter connectivity in CR studies? Yeah, we're taking that very seriously now. There's two ways to think about it. One is, in terms of the efficiency and capacity of networks, one could really think of these DTI measures as something that might really connect areas and really influence efficiency, capacity, and we're looking at that. The other thing is that I think the whole cognitive reserve claim is that we're seeing something over and above just brain limits. So the more we understand how these networks work, the underlying basis of resting bull, DTI, are the things we can measure going to identify everything, or is it just going to be that our exposure is really influenced, let's say DTI in some way, or can we see utilization of networks over and above what our structural measures provide us?
Okay, so there's another one. What do you think about the role of diffusion imaging in CR studies?
That's what I think I did. One question, is there evidence of how cognitive reserve differs in healthy aging compared to dementia? Yeah, I mean, it's really interesting. When I first started, the first observations that we made were in Alzheimer's disease, and my first imaging studies were in Alzheimer's disease. I didn't publish that. I didn't show them here, but the very first cognitive reserve 015 PET studies we published were in Alzheimer's disease. But then it became clear to me that the old people were having trouble with this task compared to young. Yeah, I mean, I think that the mechanisms and the ideas are exactly the same. I think that what we add in, with Alzheimer's disease, and now we can really quantify it in a way we couldn't then, is now we can really measure the underlying pathologies. We can measure tau, we can measure amyloid burden, et cetera, so that we can really look at that. But I think the basic ideas are exactly the same.
Okay, so there's another one. What do you think about could you comment on the difference or similarity between neural efficiency and cognitive reserve?
Right. So I think that neural efficiency is one of the ways that I feel that cognitive reserve might be implemented. So that, people find these phenomena, so I think that, and that's what I would call that part of the neural reserve, efficiency, capacity, but it's not separate from cognitive reserve. But, I had this conversation with Roberto Cabeza. He said, well, what's the difference between cognitive reserve and compensation? And I said, well, I think compensation is one way that reserve might be implemented. And the same thing, so people can study compensation without paying attention to reserve. That's absolutely fine. But differential compensation might underlie reserve. And I would say the same thing about neural efficiency. It's another way that reserve might be implemented. And there's probably multiple ways that reserve is implemented. Do you think multiple sclerosis and stroke are good for evaluating reduction in CR over shorter periods of time? Yes. And actually, there's a few people doing reserve-related work in MS. Jim Sumowski, he's now at Mount Sinai, and Vicki Levitt, who's in our group. MS is a very interesting disease in that it's affecting young people, and on an epidemiologic level, Jim has found very much similar things to what we found. Given a certain amount of disease burden, people with higher reserve exposures are doing better, and he's managed to show some some cognitive domains that seem to be more like brain reserve and susceptible to brain reserve. Just the size of the brain makes a difference, whereas others, it's more like a cognitive.
Okay, so before we go ahead with those text questions, is there anybody who would like to pose a question in person? So please go ahead now. Not currently. Okay, yeah, Yaakov, you're welcome to go ahead.
Okay.
Oh,
okay. There we go.
I have one question. In your experience and all your studies, have you observed difference in the different domains? Speaking about cognitive reserve, it is a global effect, or you could determine that cognitive reserve is more appreciated in memory or in executive functions or in speed processing?
Yes, that's a very good question. I think there might be differences. You know, there's different ways to think about it. So most of our earlier studies, like as I said, were focused on task-related activation. So what we were finding was very much particular to that task. You know, we have an understanding of the neural networks, let's call them, or fMRI networks that underlie that task, and we can see differential utilization of those networks as a function of reserve exposures. The shift over to sort of task invariant networks or some resting bold pattern starts to think about something that's more generally beneficial. But I still think it's possible, even on that level, that it might work differently for different kinds of cognitive functions. You know, that's why at this point we're measuring multiple domains. Yeah, but I don't know the full answer to that. But I think that when you really get down to the nitty gritty, every cognitive domain has its own particular demands. And it could be that people develop reserve in some specific areas that are specific to their experience, for example, as a very simple example. So you're asking a complex question, and we don't know fully the answer.
Cool. Anybody else who would like to come forward in person?
Yes, I would also like to ask a question. Yaakov, great talk. I think it would be also interesting to see how cognitive reserve is built up, and for that we would have to look into younger people and maybe even children. So do you have a strategy? I would be very keen on hearing your opinion of how to go about to study cognitive reserve in younger people when there is no brain pathology.
Well, so in the studies that we're doing now, I don't have youngsters. We're doing people 20 through 80. And now we're doing follow-up, so there it's 25 to 85. So we certainly see these things applying to younger people who clearly have no pathology. But I think you're talking more about on the child development level. Yeah. I think that would be important. You'd have to sort of decide where you wanted to start. I think on the epidemiologic level, we could do that a little better. So the guy that I'm most jealous of right now is Marcus Richards, because they have this British birth cohort, these people, you know, the 1946 cohort. They know everything about these people from the time they were born, even before they were born, and now they're imaging them. You know, so they didn't start imaging them till they were in their 50s, 60s, but still they really, so at least they can start to look at the kinds of things that you are talking about in a very informed way. Could you do serial imaging studies of some sort? Yeah, I mean, I think that's another way to go, but certainly a lot of the key exposures could be very early in life, and it would be really interesting to see how they emerge. Yeah.
Thanks. Yaakov, there was another question on sex differences in reserve.
Right. That's a very good question. I have to say, I could give you two answers. So far, we haven't noted any. On the other hand, I cannot say that I have systematically looked at it. And I'll take a little sidebar. We did an exercise study in younger people from 20 through 60, 20 through 65, basically just a straight aerobic stretching, aerobic exercise versus stretching toning. We found an effect of exercise on cognition, particularly executive function, that was moderated by age, such that older people seemed to improve more than young, but young people improved as well. And the older were young for an exercise study, we found increase in cortical thickness, all of that. Then we went back and looked at the results by sex and found out very strong sex moderation, such that the men were benefiting much more than the women, no matter what their age. And then we looked back at, some people had thought they had looked at it, but in these meta-analyses, all they did was they looked at studies that were primarily men or primarily women and said, oh, those must apply to women, but they hadn't. So we're actually going back and getting the true sex-wise data from studies to do a meta-analysis on this. So it's a very strong cautionary tale that we really have to go back and very, very carefully look for these sex differences. And I can't tell you definitively that I've done that.
Yeah, that's a good point. So, often we just control for sex, and rather than assessing explicitly the possible interactions with sex.
Yeah, I mean, I was really quite surprised. It's so interesting because the journal that the exercise paper itself was published in Neurology. Then we went back to them with the sex difference, and they weren't even interested in reviewing it. And I thought that, wow, this is much more important news than the exercise effect, which is good, but sort of expected. Yeah.
Yaakov, there's another one. So what about neural efficiency? Could that be thought of as a flexibility in changing between brain states, and could that be potentially addressed by looking at variability in dynamic connectivity?
Yeah, I think so. I think that that's true. We can. What I think we need, I sort of hinted to it on my conclusion slide, is we need to understand our tasks very well. So I had a graduate student who did a very nice study using matrix reasoning, like the Raven's progressive matrices, because it turns out that you can classify matrices by the best strategy to solve them. So we're still working this up for publication, but he was able to show that there were differences in people's ability to switch between these strategies, which I would call a very basic measure of flexibility of solution strategy. So some need a logical strategy, some need a visual strategy. You can create very simple matrices that do that, and he can look at differential ability to shift between them. And we could see that that was related, we thought, to some proxies for research. So let's say we really understood this, the cognitive demands very carefully, and then we could map that alternation in solution strategy back to brain in a dynamic way. Then we really have a wonderful tool that's much more useful than this heavy-duty hammer that I'm saying of just making a task harder and harder. It was like a lot of the tasks that I presented to you. So my point is that the cognitive neuroscience, really good cognitive neuroscience is dependent on good cognitive tasks and good understanding of our tasks, how they work, how their underlying activation, and then we can really do what the person who's questioning has suggested, I think.
Mm-hmm.
Julia, right? Okay.
Okay. Yeah, is there anybody else who would like to ask a question? No?
Yes, I would like to ask a question. I have written it in the on the side, but that doesn't matter. I was wondering whether the amyloid deposition whether it was mapped, the amyloid deposition and the areas of cognitive reserve, whether there is a kind of a superimposition or whether they are not correlated at all.
That's an interesting question. So it's a complex question. So it's about amyloid deposition, and does it specifically impact, maybe you're asking, on areas that might be part of some reserve network. So again, I'll deviate a little from your question to just back it up. So if you remember, Randy Buckner, when we were first started talking about the default mode network and the nodes of the default mode network, was trying to show that the areas where there's greater activity of the default mode network also seem to be, at least visually, the same areas where amyloid is accruing, is aggregating. Bill Jagast wrote a paper sort of like that too, where there might be activation hubs, and for people who utilize those hubs more, amyloid might be depositing more. So it's just to back up and say there could be some relationship between amyloid deposition and utilization of areas. One might posit then that if someone had greater reserve, those areas might be more efficient and less susceptible to amyloid deposition. We're looking at that now. That's why I can talk about it. We're thinking about that. I think the other way that you're talking about it is, I think for what you're asking, we would need more generic, let's say, some frontal connectivity network like Nico and Michael have written about. And is that in particular disrupted by amyloid or tau deposition? Yeah, but I think it's an interesting question to think about. What we've mainly done is just use amyloid and tau deposition as covariates to see, as people have a little more amyloid, what happens? Do we see less efficiency in tasks? Do we see less capacity, etc.?
Thank you.
You're welcome.
So another question was, Yaakov, do you see a link between hearing loss or vision impairment and cognitive reserve? This possibly relates to that recent Lancet Commission paper that found hearing loss as a major risk factor of dementia.
Yeah, honestly, we haven't looked at it. Yeah, I mean, look, I don't know if I would relate hearing loss to cognitive reserve, but certainly hearing loss does make another demand on the cognitive processing. I mean, that's been well known for quite a while. And so I think careful hearing measurements might really allow us to look at that more carefully. It could be that some people are more resilient in the face of hearing loss than others. So I don't think that hearing per se is a reserve measure, but I think it's another change that we have to cope with. That's not a brain change, but more a sensory change. You could start to think about visual loss the same way.